Hello, good afternoon, everyone. So, let me introduce myself. I'm Dr. Joita. I have been working in ISM for last 7 years, and I will be taking your physiology classes in the ISM colleges. So, feel free to ask me in case you have any doubt. You can just write through the chat box. You can communicate with me. So, this is a first physiology orientation class that we are going to have, right? So we start with the first topic, that is membrane physiology. Now, what is membrane physiology? Membrane physiology, it refers to the, this is like refers to the membrane of the cell. Okay. Now cell, as we know, from your sixth standard itself, you have studied biology. And when you have studied biology, you are quite aware of the term, aware of the structure, that is cell. So how do we define cell? As how, what has been the traditional way of defining cell? Cell, it is the structural and functional unit of life. Right? So whether it is a plant or whether it is an animal or it is our body own itself, we always have the structure that is called the cell. Now cell, the outer covering of the cell is cell membrane. Now the cell membrane, if I am drawing this as a human cell, okay, the outer covering of the human cell, this is the cell membrane. Now, the cell membrane plays a very important role. Cell membrane has a role as like covering the cell not only for its physiological function, but also it protects the cell. And it is through the cell membrane that different type of, various type of interaction and transportation happens to the cell. So starting with the cell membrane, we have to understand the composition of cell membrane. That is the first topic that we are going to start with. So anyone, do you know what is the cell membrane made of? What are the structure? What are the ingredients or components of the cell membrane? What is it made of? Yes, you can tell. What is the cell membrane made of? Cell membrane comprises of two main components. Okay, what are the two main components of a cell membrane? One are the lipids and another one are the proteins. Okay, now how this lipid and protein are oriented in a cell membrane? Is it that like there is like layers of just one layer of lipid, one layer of protein? Is it like that? No. Now when studying of this cell membrane happened, when they started studying the cell membrane, many theories came into existence, like the theory for the cell membrane structure. It is multiple. First structure, there was like one sandwich model also of the cell membrane, which says that the protein was in the middle and there were like layer of lipid on the either side. But that became obsolete. That was totally ineffective and that could not satisfy the function that the cell membrane was carrying out. Then gradually there was a triple layer theory also. Like there is a triple layer of lipid and protein, but that was also not proven. Over time, it was the ultimately, ultimately the fluid mosaic theory was accepted. Okay. Now, what does this fluid mosaic theory proposes? This fluid mosaic theory, it says that according to the fluid mosaic theory, the protein molecules are interspersed, like that of a fluid, like that of a mosaic floor. You see the background is like white in color, and then there are like that mosaic which is like interspersed every here and there. So the protein molecule are interspersed in the lipid bilayer. Okay, and according to that, they have also like postulated a diagram, a configuration of the cell membrane. So if we draw the cell membrane, it will be like this. We'll just draw a cell membrane for our understanding. So this is our lipid bilayer. I'm just making a linear diagram of the cell membrane. So this is like the layers of the lipid, and then there are this protein molecules that are interspersed. Some lie across, some lie sticking to the ECF, and some lie sticking to the ICF. This is like the overall structure. Now, this structure, which we call as the lipid bilayer. So see, whenever any substance, whether it is water or whether it is something else, when they try to enter the cell, the first barrier, the first structure that will come across is this cell membrane. Now, this structure, this globular head, it is nothing but the lipid bilayer. So, how does a lipid bilayer look? So, this is a lipid bilayer. Bilayer means two layers. Now, in this lipid bilayer, the structure that we have is a phosphate globular head, right? This is a phosphate globular head, and underneath, below this, what we have is a fatty acid tail. Okay, fatty acid tail. Now, chemically, if we study further this bilayer, okay, we take out like one of this phospholipid, it stands out to be something like this. Okay, it stands out to be something like this. Okay, where this is the phosphate head and this is the fatty acid chain. Now this phosphate head, it has got a phosphate group. It has got a phosphate group and a glycerol background or a glycerol backbone. And on the tail part, we have fatty acid with saturated and unsaturated fatty acid. Now, it is this globular, it is this globular head which is hydrophilic in nature. Okay. Now, this globular head is hydrophilic in nature because it has got a glycerol background and it has got a phosphate group. Whereas the fatty acid is hydrophobic in nature. Now, this globular head is hydrophilic and the fatty acid is hydrophobic. So, since this molecule, this phospholipid, it has got, since the phospholipid has got both hydrophilic and hydrophobic region, it is called as amphipathic. It is called as amphipathic molecule. That means it has got both hydrophilic and hydrophobic part. So it is amphipathic in nature. The cell membrane is amphipathic in nature. How did we find out that it is having both the hydrophilic and hydrophobic region area? It is seen that when this cell membrane is added to a water oil interface, when it is like a drop of this in this water oil interface, the cell membrane are added, it is seen that the phosphate region aligns towards the water and the fatty acid area aligns towards the oil. So, like if this is a water oil interface, so this is the water and this is the oil. So, what we get to see is that towards the water, all this globular head aligned, and towards the oil, this fatty acid tail, it aligns. This is how it looks. Okay, this is the structural view of it. Coming to the physiological aspect of it. How and why should we remember all these things? What is its importance? Its applied aspect of cell membrane. See, cell membrane is something that you have studied from your school days. Then why all the way when you come to study medicine, you have to understand the or know details about the cell membrane? Very simple, because all the essential things that gets inside the cell has to pass through the barrier of the cell membrane. Therefore, to understand the structural integrity along with the function of the cell membrane is very, very important. The applied aspect of the cell membrane lies in understanding the permeability of substances across the cell membrane. Now, permeability of substances across the cell membrane obviously will depend upon which substance is more soluble in water, or how much is it soluble in the lipid, right? It is seen that like hydrophobic substances. What are the hydrophobic substances? Like oxygen, like carbon dioxide, means the substances which do not dissolve or which is not very friendly to water. These are the hydrophobic substances. But hydrophobic substance also means like lipophilic, like they don't like water but they like oil, means the lipid portion of it. So hydrophobic substances like oxygen, carbon dioxide have more permeability across cell membrane. Whereas compared to that, obviously hydrophilic substances like water, like amino acids, they have less permeability. This is one of the applied aspect of the cell membrane. But when a substance has to pass through the cell membrane, pass across the cell membrane, it does not only depend on how much it is soluble in this water portion, like water soluble or lipid soluble. Other factors like other factors affecting the permeability are there also, like size of a molecule. Okay, after that we will come to the constituents of the cell membrane in detail. The size of the molecule, what is it? Like if a molecule is having less diameter, then it passes through the cell membrane easily. Okay, if it has less diameter, it passes through the cell membrane very easily. Another point is that the factors like partition coefficient, diffusion coefficient also determines permeability. Okay, like partition coefficient means how much a substance is more soluble in the lipid compared to that of the water. If the partition coefficient of a substance is more, then obviously the permeability will be more. Diffusion coefficient, if a substance has capacity to diffuse more across the cell membrane, then again the diffusion or the permeability of the substance across the cell membrane will be more. Okay. Now moving on to the other parts of the membrane transport, like before that, let us talk about the types of the lipid and the protein that is present in the cell membrane. Why we have to do so? Because right after that, when we discuss the pattern of transportation across the cell membrane, both the active transportation and passive transportation, then it will be helpful for us to understand the types of lipids that are present in cell membrane. What are the types of lipids that are present in cell membrane? Okay, they're mainly of like phospholipids and glycolipids. Okay. Now, one more point in context to this lipid and protein, please do remember in almost all cell membrane, the protein is more than lipid. But there is only one exception, except the myelin sheath, except the myelin sheath. Now, there are two types of lipid. One is a phospholipid, another one is a glycolipids. Now, what are the types of the phospholipids? Phospholipids are more than one. These phospholipids, they are of the type phospholipids, they are of the type phosphatidylcholine, which is lecithin. Then we have phosphatidylserine, which is also abbreviated as PS. Then we have phosphatidylethanamine, phosphatidylethanolamin. Okay. Then we have phosphatidylinositol, which is the precursor of secondary messenger. Then we have the sphingomyelin and also another important phospholipid which is present in the inner surface of the mitochondria and is called cardiolipin. Now, in context, just for our exposure purpose, in context to all these components, we will just mention the name of one clinical condition. Okay. Phosphatidylcholine lecithin. It is very much essential for the maturation of lungs in a fetus. So, the clinical condition associated with deficiency of lecithin means if lecithin is not adequate during the pregnancy in the lungs of the fetus, it will lead to respiratory distress. If phosphatidylserine is not present in sufficient quantity, it leads to a hemorrhagic like bleeding disorder which is called as Koch's syndrome. Just know the name of the diseases. Phosphatidylethanamine or cephalin, if this is inadequate, it is seen to result in certain, like indirectly, not directly, but indirectly, again, bleeding disorder. Okay. And phosphatidyl inositol, it is a precursor of IP3 and DAG. Now, this IP3 and DAG both are very important for the contraction of smooth muscle. So, in efficiency in muscle contraction. Sphingomyelin, if the enzyme sphingomyelinase is inadequate or missing, it leads to a congenital disorder, which is Niemann-Pick's disease or syndrome. Cardiolipin, if the gene is mutated that makes this phospholipid, then it leads to a syndrome called Barr's syndrome. So this is just example of few diseases that we should get used to in context of this. This is our lipids. Now coming to the protein part. Just like the lipids, protein are also very essential component of the cell membrane. So proteins are of two types. Okay, proteins are of two types. Some protein, they are like integrated. They are like within the cell membrane. They lie within the cell membrane and it traverses like all the way across the lipid bilayer. So orientation of some protein are like this, you know, like this. They are like spanning, they are like touching both the ECF as well as ICF. So such type of protein are called as transmembrane or integral protein. Okay, whereas there are certain proteins which are like sticking only towards the ICF or they are sticking only towards the ECF. So such are called as peripheral protein. Now, what is the physiological importance of this peripheral protein and this transmembrane protein? This transmembrane protein are for transportation, are for transportation. Like they act as, you know, carrier protein, they act as channels, they act as ports, all this passages they act as. And this, they also span the cell membrane. Okay, they also span the cell membrane. Now, example of such protein are sodium, potassium, ATPase. It's a very beautiful example, sodium, potassium, ATPase. Then, coming to the peripheral protein. This peripheral proteins are for providing structural support. They will give the strength and mechanical stability, are for the mechanical support, that is structural stability to the cell. Okay. Example, spectrin, ankyrin in the RBC. Now, this protein, what they do, they RBC, it is not a round in structure. It is a horseshoe shape. It is like a biconcave shape. So, this pectin and ankyrin, they kind of supports the structure of the RBC. They are the structural supporting protein of the RBC. Okay, these are the components of the cell membrane. Now, as I have told you just before this, we need to, all this thing is in context to the primary function of the cell membrane, which is the transport across the cell membrane. Now, what are the different type of transport across the cell membrane? Transports across the cell membrane. What are the different type of transport across the cell membrane? Now, the physical transport, all the types of the transport that is there in the cell membrane, they are they can be broadly classified into two types. One is the passive transport, okay. Another is the active transport. Now, in passive transport, what is a term? It is called the passive transport. Passive means here no energy is needed. Now, body is constantly making this energy in form of ATP. So, whenever there is no usage of ATP during this process, during this transportation, it is called as the passive transport. Okay. Whereas in active transport, ATP is needed. ATP is needed. The examples of this passive transport, is osmosis and diffusion. So, osmosis and diffusion are the two examples of the passive transport. In this transportation, no requirement of ATP. Whereas active transport is again of two types. Primary active transport and secondary active transport. Okay. Now, this primary active transport and secondary active transport both needs carrier as well as ATP. Secondary active transport is further divided into co-transport, that is when transportation is taking place in the same direction, counter transport and uniport. Counter transport means when the transportation of two substances are happening in the opposite direction, and uniport means when the transportation is taking place only in one direction. So, moving on to the type of the passive transport. So, we begin with passive transport. So, if I am drawing this cell membrane, say this is the cell membrane and this is the ECF and this is the ICF. Okay. Talking about first diffusion. We'll start with diffusion, then we will move into the osmosis. Then in our cell, we have got in our cell, it is surrounded by the body fluid. In our body, we have got various molecules that is floating, that is in the circulation along with the blood. Okay. Now, they are like different kind of substances. There are amino acid, there are these cations, anions. Body comprises of like cations, anions a lot. Now, outside the cell and inside the cell, there are presence of positively charged particle as well as negatively charged particle, but in different concentration. So, say for example, there is a particular molecule which is outside the cell, across the cell membrane. Okay, and its concentration is more across the cell. So, this is the cell membrane and its concentration is more in the ECF compared to that in the IC. So, this difference in the concentration of this difference in concentration of the substance will create a gradient. Will create a gradient. So when this gradient means when the concentration is different outside the cell and inside the cell. So this gradient, this concentration gradient will cause just passive movement of this substance. Like this particle will just move across the cell membrane into the ICF, just without the need of any energy, without the use of any carrier protein, any transmembrane protein, any carrier protein, nothing will be needed. Just following the concentration from a region of from region of higher concentration to lower concentration. Okay, just simply like that without the need of energy and carrier, the substance will flow from ECF to the ICF because in the ECF the concentration is more. This is called diffusion. Now in diffusion there are two types, simple and facilitated. Now this is an example of simple diffusion. This is an example of simple diffusion. So if we represent this one in a graph, how will it stand out? It is like, so this is the graph to show the simple diffusion. This is the concentration, and on the x-axis we have the rate, and there we have y, we have the concentration. So you see, as the concentration increases, there will be linear increase in the simple diffusion, meaning as long as the concentration of molecule is more, the rate of diffusion will be higher. This is an example of simple diffusion. Now, how is this used in human body, in our system? In our human body, the example, physiological mechanism that follows this one is the diffusion of oxygen, carbon dioxide across the respiratory membrane in the alveoli. Okay, the respiratory membrane in and across the alveoli, when the gas is diffusing, it follows only the principle of simple diffusion if the person is healthy. If the person is healthy, this is an example of simple diffusion. Of course, if the person is unhealthy and there are other factors, then of course this simple diffusion will be affected. These factors and all we will discuss gradually as we do the systems. So this is a classical example that follows a simple diffusion in human body. Next is our facilitated diffusion. So what is facilitated diffusion? Facilitated diffusion, here a carrier protein is needed, but no energy is needed because it's a passive process. So here the classical example of this is transport of glucose by glut to protein, by glut to protein. So the graph, if we draw for the facilitated diffusion, stands out to be like this concentration and this is the rate. So in case of facilitated diffusion, what do we see? With the increase in the concentration, the rate of diffusion increases, but after some time, it becomes static. There is no diffusion even when the concentration is increased. Now this flat portion, the plateau where it begins, this is called the Vmax. Now, why, unlike the simple diffusion, we get this kind of a hyperbolic curve for the facilitated diffusion? Why? Because of the limitations of the carrier protein. Factors affecting facilitated diffusion. So, factors that are affecting facilitated diffusion are the saturation of carrier protein. Now, it is seen that the binding site, like the carrier protein, it has to bind, right? Say, for example, if we draw out the cell membrane, okay, we draw the cell membrane, and just for our understanding sake, let this be the carrier protein GLUT2. Okay, draw it in a simplified way. This is the cell membrane, and say this is the like carrier protein GLUT2. So it has got a binding site. So on its binding site, what is binding? It is the glucose molecule that is binding, right? It is the glucose molecule that is binding. So once this glucose molecule is bound, what happens is a conformational change. What happens is a conformational change which later on delivers the glucose in the ICF, ECF to the ICF. So it is seen that this is the binding site, this is the binding site. So it is seen that after certain period of time, the binding site gets tired out or gets exhausted or saturated after transporting for some time. For some time. This is one of the reason, which is the saturation. Second reason, it is the stereotype. Stereotype means the binding site on the carrier protein, it will transport and bind and carry only one type of the optical type of the glucose. Like GLUT2 will transport only the dextro, the D-glucose isomer, only the D-glucose isomer. It will never transport the L-glucose isomer. Third is the competitive inhibition. So it is seen that for the binding site on the GLUT2 transporter, galactose, like D-galactose rather, galactose competes with the binding site for glucose on the transporter. Okay. So galactose, it competes for the binding site on the transporter. These three factors are the reason why the facilitated diffusion cannot go on or have a linear graph like that of the simple diffusion. This is the diffusion with an example. Next, what we have is the osmosis. Now, osmosis is also a passive process, but here it is actually movement of water molecule from a region of low concentration to a region of high concentration. Okay. So it is actually the movement of the solvent. It is, or rather we can tell it is like the passive diffusion of the water. It is a passive diffusion of the water. So say for example, like this is a osmosis is a process that all of you know. You have studied already in your school days. So see, suppose this is a vessel which has a permeable membrane. Okay. So this is A and this is B. So where will the movement of the water particles be? The water particles following the laws of the osmosis, what will happen? It will move. So here this round structures are the solute, rest is the solvent. So here you see in compartment B, there is less molecules or the less solute, right? So ultimately here this is the region where the concentration of solute is like less. So ultimately the water will move from B to A. This is osmosis. It is the movement of the water from a region of low concentration to a region of high concentration of what? Of the solute. Wherever the solute is low, from that concentration to the region where the solute is high, to that concentration, there will be movement of water. Now, how does it physiologically implicates in our body? What is the physiological implication? So, fluid or water always moves it always moves to hyper osmotic region or solution from hypoosmotic solution. Okay, this is how it implies in our body, osmosis and diffusion. Coming to the secondary transport. Now, secondary transport or secondary, let's start with the active transport. That way we understand that better. Active transport, which is the primary and secondary transport. Okay, primary transport and the secondary transport. Now, a very classical example of the primary transport, that is what we are going to use to understand the primary active transport, is the sodium potassium. We'll understand with the sodium potassium ATPase pump. Okay. Primary active transport. As we studied, active transport means what? There is usage of ATP. There is need for the energy. Now, the example that we are going to study is with sodium potassium ATPase. Now, this sodium potassium ATPase or sodium potassium pump, what are the characteristics of it? First thing about this sodium potassium pump, we have to remember is it is abundant in all cells of the body. Whatever cells we take, whether it is peripheral cells, nervous cell, everything, everywhere this sodium potassium pump is there. And sodium potassium pump is essential for various functions of the cell to maintain the electrical neutrality of the cell also. That also gradually we will study. It accounts for like 70% of energy used by the peripheral cells and about 25% in the central nervous system cells. Like this sodium potassium ATPase pump, you can see the term ATPase, that means it has an enzyme that breaks the ATP. So this pump is such that it consumes a lot of energy. Out of the total energy consumption, the body for the various metabolic function, the cells for the various metabolic function, it uses ATP. So out of that, 70% of the ATP is used by this pump alone. Okay, it is consumed by the pump alone in the peripheral nerves and 25% in the central nerve cells. Okay. Now, what is the structure of it and why it is so important? The structure of sodium potassium ATPase. What is the structure of the sodium potassium ATPase? First of all, this is electrochemical pump and it is a heterodimer. So the term heterodimer, what do we mean by the term heterodimer? Dimer means it has got two subunits, right? It has got two subunits and the subunit has multiple sites. So if we draw this one, if we draw this sodium potassium ATPase pump, it is like okay, this is the beta subunit. It's a dimer. Dimer will have two subunits. This is the beta subunit and this is the alpha subunit. The structures are all obviously hypothetical. This is the alpha subunit. Okay, now this subunit, they have got multiple structures in it. Okay, the beta subunit, it does not have any binding site as such. It just has three sites for glycosylation. It is the glycosylation site. Okay, site for glycosylation. It is the alpha subunit that has got multiple binding site and it is like little bit more compared to that of the beta subunit that goes in the cytoplasm, that touches the ICF. The alpha subunit, it is touching the ICF more prominently. So here it has got a sodium binding site. Okay. Apart from sodium binding site, from towards the ICF, it also has got a catalytic site. Okay, catalytic site means it will break down the ATP, or we can say it is the ATP breaking site. Okay, then we have the phosphorylation site, like where the phosphorylation phosphate group will be added on. Now here the side facing towards the ECF, it has got the potassium binding site, and very close to the potassium binding site, what we have is the covalent binding site. There are like covalent binding site. So the main function of the sodium potassium pump is to, it pumps out 3 sodium molecule from ICF to ECF and pumps in 2 potassium molecule from ECF to ICF. Another important function is that the digitalis or decoxin, like drugs like digitalis or decoxin, bind on to the alpha subunit, that is a covalent binding site. Okay, to treat cardiac conditions, like in a failing heart, this is used to improve the beating of the heart, the contraction and the beating of the heart. So this is a very classical example of the primary active transport. Okay, coming to the secondary active transport. Now this secondary active transport. Now secondary active transport, as we saw, it is of multiple types. In secondary active transport, usually what happens? Usually, not only, okay? Usually, here, one molecule is transported with glucose. Okay. Now, with glucose, it can be in the same direction or in the opposite direction. So, if we draw for our understanding benefit and try to understand the thing. So, this is the ECF and this one is the ICF. Okay, we are just doing it inside the box so that we can understand it better. This is not the cell membrane. This is the cell membrane, which I am just drawing in a single line, and say this is the ICF. So substances, there are multiple substances. Some substances may get like symptoted, or together they may enter the cell along with glucose. Okay, so it may be something like this, like sodium and glucose symport. So here you see sodium and glucose symporter, which is a co-transporter. Here both the molecules are entering from ECF to ICF. Okay, this is a sodium glucose symporter. Similarly, other substance that enter the cell along with sodium, as seen in case of a thyroid follicle, is the sodium iodine transporter. This is also symporter, co-transporter. Okay. Another transporter, which is sodium calcium exchanger, very commonly seen in the muscle, especially the cardiac muscle. Very important. Its function is very important in the cardiac muscle. So here what happens actually, here three molecules of sodium enters, three molecules of sodium enters and one molecule of calcium leaves. So it is a exchange. It is like not a co-transport. It is like a exchange. It is a exchange between the sodium and the calcium. Here, three molecules of sodium is entering from ECF to ICF and one molecule of calcium is leaving ICF to ECF, and this pump is very important after contraction of cardiac muscle happen and the muscle has to come back to its resting position. These are the typical example in which sodium is essential. Now there also lies another exchanger which is not dependent on sodium, but it is a very good example of the counter transport. It is seen in the RBC and also in the kidney cells, in the renal tubular cells. Like here, it is the exchange between the chloride and the bicarbonate. So here the chloride enters and the bicarbonate goes. It is also called as the anion exchanger. So these are the example of the secondary active transport. Now, if this transportation is like mutated, just understand, get exposed to the clinical condition that might happen whenever there is any mutation to any of this transporter. The clinical implication, why we should know and what might happen, like talking about the clinical implications. Say for example, our first thing, that is the sodium potassium ATPase. Okay, that is the example of the primary active transport. Clinical example I am writing now. So if this pump is mutated or inadequate, okay, it will affect the metabolism and electric balance of all cells in the body. There'll be a huge problem maintaining the electric neutrality, also the metabolism, because sodium potassium pump has to constantly pump and maintain the membrane potential. This is very important. Okay, this is what will happen if this primary active transporter is affected. Second example, see, this is the first example, this is the second example. Say the sodium glucose symporter is affected or mutated. Then what will happen? The person will suffer from malabsorption of glucose and galactose in the enterocytes, ileum enterocytes. There will be problem, the transportation will be affected. There will be problem with absorption of glucose as well as galactose. Third example, sodium iodide pump. So, if this is mutated or affected, then the person will suffer from congenital hypothyroidism. Just a very small mention to you why this will happen, just for you to remember and understand. Actually, how this sodium iodide pump it acts. So this is the thyroid follicular cell. So here this sodium and iodide symporter acts. So by this sodium will get in and iodide will also get in. Later on this one will combine with the other iodide and form iodine. Now this iodine will be used to synthesize the thyroid hormone. So if this pump is mutated, if there is any defect in this pump, this transportation will not happen. So the concentration of iodide will be inadequate. So iodine will also be less and less and less in quantity. So without iodine, how can there be formation of thyroid hormone? So eventually the person will suffer from hypothyroidism. The fourth example, as we mentioned here, the anion exchanger, which is the chloride and the bicarbonate pump, right? What will happen? That will, if this exchanger is mutated. So what did we study? It is most evident in the RBC, right? In RBC, this exchanger forms band 3. Okay. So the RBC functioning will be affected. Okay, so these are the examples of the secondary active transport and its implication if it is not working properly. Another thing in context to membrane physiology that you have to remember is the membrane symmetry, after which we will discuss the membrane fluidity. Okay. Now, membrane symmetry. All this while, what did we study? We studied the lipid bilayer and the proteins are interspersed. That structure we studied. Now, this membranes of the cell, it is not always linear. There are certain region. There are certain region in the membrane. Like there are certain region in the membrane which are called as the lipid raft. Okay, certain region in the membrane, they are termed as the lipid rafts. Lipid rafts. Now, these lipid rafts, they can be linear or they can be in the form of invagination, which is called the caveole or caveole. So, the lipid raft, which is like linear, some region in the cell membrane which is like linear, it will be like this. Okay, whereas the caveolae, they will be like something like invagination. There will be some invagination like that of a pot, like that of a pitcher. This is called as a caveole. Okay, and the protein that is involved in it is called the caveolin. Now, the linear one that has the lipid raft, that has this lipid raft. Now, this lipid raft, what are they rich in? It is seen that this lipid raft, they are rich in lipids and sphingolipids. Okay, but some of them are linear, like some of them are linear in structure, and some assume this in vagination. Now, this structure, which is the caveolae, this is essential functionally. Why is it necessary? It is seen that folate gets into folate gets inside the cell through caveoles. Okay, it is through the caveoles that folate, which is essential for incorporation and maturation of the hemoglobin in the blood, it is entering the cell through this. And not only that, SP40 papilloma virus also enters the cell through the caveoles. These are like the applied part of the physiology in context to this lipid raft. Lastly, we will talk about the membrane fluidity. Now, there is this membranes, like membranes are like gel in structure. Okay, these membranes are jelly-like structure, gel in structure at body temperature. At body temperature. Now, there is a temperature which is called the transition temperature. At transition temperature, the gel becomes more fluid and vice versa. So see, there are like various conditions of the body. Sometimes the body is like exposed to high temperature. Sometimes the body is exposed to low temperature. So all this temperature ultimately affects the jelly structure or the fluid structure, the more like, the state of the cell. Okay. So, transition temperature is the temperature at which this cell membrane surrounding the cell, it becomes more jelly, little bit more jelly-like or little bit more fluid-like. So, it is seen that at higher temperature, the cell membrane becomes more gel, and at lower temperature, the cell membrane becomes more liquid. Okay, this is a transition. And in all this, it is the saturated and unsaturated fatty acid which affects the transition. Okay. The other component of the cell which is affecting this one or maintaining the fluidity or the gel like is the cholesterol. So cholesterol, it acts as membrane buffer. It acts as membrane buffer. So it is the cholesterol which is placed like between this lipid bilayer, which is interspersed between the lipid bilayer, is acting as the membrane buffer and maintaining and keeping the gel or the liquid-like configuration of the cell membrane. So students, in today's class, we had the orientation of the cell membrane. First, we started with the components, the details of the components, and the applied part of it, that is a permeability, and the functional part of it, that is a transport system. So the next orientation class, we would be taking another topic and going like details into it. So tomorrow I will not be available. Our next orientation class will be day after tomorrow, that is on Thursday. In case of any doubt, you can always communicate to me through the chat. Okay. So I will see you all in the next session. Until then, bye and thank you.